Hi, I'm Andrea. Welcome to today's Harper Lecture. The Harper Lecture series highlights the University of Chicago's collective commitment to the highest aspirations and standards in research and education. Today, you will hear from our world-class faculty and enjoy stimulating conversations on critical topics, all in the comfort of your home.
We extend our appreciation in advance to our global university community for their collective efforts in supporting the mission of the university in today's deeply challenging environment. Technology has allowed us to remain connected and engaged, and tonight, you too will be part of the conversations. A couple of notes. All participants will be muted. However, we do encourage you to ask questions. We will have plenty of time for questions at the end of the presentation. If you have issues with audio, you may want to shut down your programs running in the background or dial in from your phone.
Hi, I'm Colin. On behalf of everyone at the UChicago Alumni Office, we offer our deepest gratitude and appreciation for your continued support. We're proud to be part of this community as it has come together during these trying times to invest in our students, empower our faculty and scientists, and extend care and connection. And now it's my pleasure to welcome you to the 41st season of the University of Chicago's Harper Lecture Series. Thank you for being with us. Enjoy.
Good evening. I'm pleased to welcome you this evening to this Harper Lecture that will be on the topic of COVID-19 and beyond, understanding vaccine adjuvants and innate immune responses with Professor Aaron Esser-Kahn. I'll introduce him in a moment to you. So we're also appreciative for Alumni Relations and Development for inviting us to be with you this evening in this Harper Lecture. Aaron, if you could bring up the slides. I'd like to begin by introducing you to an aspect of the University of Chicago's Pritzker School of Molecular Engineering called the Chicago Immunoengineering Innovation Center. My name is Jeff Hubble. I'm the Eugene Bell Professor of Tissue Engineering in the Pritzker School of Molecular Engineering, and also one of the co-leads of the Chicago Immunoengineering Innovation Center. As we built up the Pritzker School of Molecular Engineering, we focused our hiring in the biological domain on immunology, the immunology engineering interface, which includes the cancer engineering interface as well. I'd just like to introduce that center to you briefly before we move on to the main show tonight, which is Aaron Esser-Kahn. So here we develop engineering approaches to address both basic and translational immunology. Next slide. What does this mean? So from basic immunology, engineering can contribute directly to even basic immunology through engineering systems to quantitative systems, systems physiology systems to address basic unknown questions in immunology, using cell and tissue engineering approaches, developing micro device tools to address questions that could not be addressed otherwise. So for example, how do you do not just one experiment, but how do you do 1,000 experiments all at the same time using microfluidic devices? How do we apply computational approaches to immunological data to understand data sets that are so complicated that they really can't be understood by the human in the absence of a computational framework? And what Aaron will discuss with you tonight, engineering molecules, molecules as both tools to probe immunology and molecules as translational aspects that could potentially be drugs, could turn them into drugs. So this is being addressed in our center in the context of cancer immunology, autoimmune diseases, in asthma and allergy, in engineering the microbiome in the gut, in the skin, in the mouth to provide for human health. And then the topic of tonight's session, infectious diseases, translation immunology to address infectious diseases. So with that brief introduction to the center that I want to describe, I'd like to introduce to you Aaron Esser-Kahn. Aaron joined us in the Pritzker School of Molecular Engineering. He's trained as a chemist So he grew up in Michigan, in the Bloomfield Hills area of Michigan. Took his bachelor's degree at the California Institute of Technology in chemistry. From there, moved for his PhD at the University of California, Berkeley, also in chemistry. And so he joined us a couple of years ago, 3 years ago, from the University of California, Irvine, to bring his unique capabilities in synthesis and design of novel molecules in the context of vaccines and other aspects of translation immunology. So with that, Aaron, the floor is yours. Thank you very much.
Oh, thank you, Jeff, and thanks for the introduction. And thank you all for coming and listening. I think this is an exciting time for vaccination, even if it's sort of a scary time in the world. We're at a really major turning point in the world of vaccination. So I'm excited I get to share some of that with you all and tell you a little bit about what my lab is doing in the context of vaccines as well, and motivate that with some of what's going on with COVID right now. So I'm going to talk a little bit tonight about vaccines, what we know about them, what important questions remain. I'm going to give a brief primer on COVID and where the vaccine world stands right now, which is changing literally every day. I woke up this morning with new news. And maybe some stuff that will be good for your Turkey Day Zoom conversations. And then I'm going to talk about our work, understanding, can we count different types of immune responses? Can we see how the cells respond to a vaccination? And then I'll tell you a little story about how can we change the conversation between vaccines and the immune system to shape it to be more safer and more efficacious. I think it probably is obvious to everybody here, but it sort of bears repeating that vaccines are still one of the most effective, cheapest public health interventions ever. And really nothing sums this up better than this infographic developed by the CDC. These are the pre and post morbidity rates after the introduction of a vaccination. And you can see that really vaccines are one of the only ways we can wipe a disease literally off of the face of the earth in some cases, which has been true of smallpox. So far, but really you can reduce the prevalence of a disease almost to nothing with the introduction of vaccination. So, but there's still many diseases on the planet, and one that's really on everybody's mind this year that don't have effective vaccinations. And the question is, what is different about some diseases than others, and how do we develop vaccines against these sort of harder to vaccinate diseases? And just, a point here is that, while we are really focused on COVID this year, I think it's important to bear in mind, just how many people suffer from some of these other diseases, particularly tuberculosis, which in many ways has all of the same challenges as COVID, but still results in a very large number of deaths every year and has for the last 100 years. So I think it's important to bear in mind that these are problems that the world has been dealing with for a long time. So there are many challenges in vaccines, and I really only work on the development of the molecular side of things. So I'm going to try and get some of the stuff out of the way before we go along, because I'm not really equipped to talk about the public health parts of this. And I'm also not really a person who can proselytize about all of the things about vaccines, but I'll just give you some basic facts and figures that I think will help. So first of all, there's this sort of myth that vaccines are somehow driving a large part of the profit. In fact, one of the main problems with vaccination is that they're the least profitable part of the biotech industry. They routinely make pennies per administration. And we'll talk about exactly why they take so long to develop. It's because nobody wants to risk the cost of development. Approved vaccines are extremely safe. They're safer than driving a car. In fact, they're actually safer than taking Tylenol, which probably you don't really think about, but there are more resultant deaths from Tylenol every year than there are from vaccination. And vaccine development, as we'll see a little bit later, is all about ensuring safety. Herd immunity is incredibly difficult to achieve without vaccination. I'll show an example of that later, and then I'll talk a little bit about sorry, I was looking at the chat there. I'll talk about COVID vaccines in particular as motivation. So this is just a great example from the website 538, and it's actually the work of Emily Oster at Brown University. If any of you guys have read Crib Sheet, she's the author of that book. It shows a correlation between the number of cases and the percentage of people, or particularly children in this case, age 19 to 35 months who've been vaccinated. And so really, in order to achieve something like herd immunity, you need extremely high vaccination rates, upwards of 95 to 99% for something like whooping cough, even to lower the rates down to this 1 to 2%. So it's really hard to achieve without complete vaccination in a population. So just bear that in mind when you're listening to people in the news right now talk about herd immunity. It's very difficult to achieve everywhere else. Is there somebody seeing captions somewhere? I don't have captions on the screen, so I'm that unfortunately the margins are simply because of the way the screen is projected, that it's just 16 by 9, so there's not much I can do about that. So the other great news that's been coming up recently, people have probably seen the cover of the New York Times and other things, is that I think we've seen really effective vaccination against COVID in recent weeks. Of course, the asterisk right now is that the data needs to be reviewed. People need to take a really good look at it, but all signs point to the fact that these are going to be extremely effective vaccination approaches, and we're really excited about that. So I wanted to talk a little bit about that and then dive into the details of how these things work. So you guys have probably heard this term warp speed bandied about. I just wanted to talk a little bit about what that meant and how vaccine development worked. So it's not really a pace so much as it is a massive public and private partnership. And speed in vaccination is not really about how fast you can do the experiments. It's mostly a financial and logistical challenge. So you can think of speed in terms of development of a vaccine as really about taking into account safety and then adding basically unlimited resources. And the difference is that this has never happened before. There's never been people willing to supply essentially unlimited resources associated with a vaccination, because, as I mentioned before, nobody is willing to take the risk to develop vaccines because they yield such low amounts of profit. Nobody wants to develop them at risk. And so this is what Operation Warp Speed really is about. It's not about taking the timeline and compressing it. It's about de-risking the timeline by dumping money into each element of the timeline. So there's a couple of things that have allowed this to happen. One of them that really doesn't get enough mention is the amount of legwork that went into developing all of these technologies. So it's not like these technologies just simply sprung into existence in March and April. All of these vaccine platforms, as they've been mentioned, already existed and had been well vetted and developed previously using several different systems. And so they were basically plug and play technologies that could be used with these new antigens and then developed very rapidly. And then really the key thing is that by combining the different phases of the process, and essentially what you're doing there is basically just paying people to de-risk each step of the process, you can basically move everything along quite quickly. And so I'll talk a little bit more about that just so that you can think about it. And really what this is basically like a Manhattan Project approach to vaccines. I think that's something that at this campus and historically we're all quite familiar with, the idea of grand challenges built on the back of large government intervention. So in a traditional development, you have 1 to 2 years where you develop all of the initial work. Around this, the difference here is that all of that initial work had already been done for analogous diseases, in this case, the original SARS and MERS. So all of that was already eliminated because those two diseases had very close similarity in terms of the proteins that were borne out on the structure of the coronavirus. So that basically chopped out those first couple of years. So again, built on the back of many years of hard labor in the NIH and many labs across the country. Then the other thing that's different is that normally in a vaccine development, you do phase 1, phase 2, and phase 3. And the real reason you do it this way is not because you need to, but because at each point here, the company is unwilling to put more money into the process until they're absolutely certain that phase 1 works, and then absolutely certain that phase 2 works, and then absolutely certain that phase 3 works, because each of these is basically an exponential amount of more money that the company has to dump in. And the difference here is that now the government has stepped in and basically said, hey, we're willing to front you all of the cost of running all of these trials. Why don't you just do them all stacked on top of each other? And as long as they work, we'll still know the same results in the same amount of time. And we'll add in production. So we'll begin producing things in tandem. And if things work, then we'll already have things rolling off the production line at the same time. So all of this has always been possible. I shouldn't say it's always been possible. What I should say is that it's been possible in the last 10 to 15 years to do this, but no one's had the guts or the capital basically to make this happen until there was this sort of emergency and the necessitated this. So I think you can think of Operation Warp Speed as the world and the United States government rising to the challenge, much like they did in the Manhattan Project, to muster scientific and intellectual capabilities to produce something that is possible with human capability and understanding. It just wasn't happening before due to the fact that the right people weren't in the right room at the right time, and the right amount of money wasn't placed before them. So that's really the difference in terms of warp speed. It's really just about de-risking the process that already existed and moving it into a compressed time frame. And for people like me who develop or think about this early stage process out here, that's mostly where I focus my time and energy. What's really exciting about this is that, you know, when you hear people who've gone through this for their entire lives, they're talking about this as the question is, will this be the future of vaccine development, or will we go back to this for every new vaccine? And if we can do this for every new disease, think about all of the lives that we could save if we simply turn this into the new paradigm for vaccine development rather than going back to this in the future. So I wanted to give a little bit of a primer about how vaccines work and what's important and what you need to know about them. And so I'll start with sort of a very simple version and then build up in complexity as we talk about them. So how do you fight a disease? I have sort of a simple version of this, and then we'll get into the molecular scale first. So you have a pathogen, you're trying to fight it. So how do you fight a disease? Well, what you try and do is cheat, right? You try and figure out how you can give yourself an advantage. So what do drugs and antibiotics do? Well, they basically figure out how to cripple the disease in some way. Either they remove some important enzyme, they damage the disease in some way. The other approach is to, the problem with that in general is that the disease usually figures out a way around that. It mutates or it grows in some fashion, and then you end up with a tougher enemy because it usually has a way around your drug. In vaccination, what you do is basically you try and make your immune system smarter. So you're trying to educate yourself in ways to combat the problem. So you make yourself smarter. You also generally make yourself tougher. These are the two basic principles of vaccination. So how do you do this? How do you educate the immune system? Well, basically you need to give it a set of information. You give it information about how to find and target the pathogen of interest. And so I have a metaphor for this that I use to explain this idea, and then we'll get into sort of the details. So I think this is very similar to sort of the wanted poster that you use for, you know, in the Old West. Basically, you give the immune system kind of the same information that you put on a wanted poster. You have the name, some sort of identifying information, then you have kind of the last known places that you look for, and then you have kind of a method that the immune system uses to find and identify the pathogen. So method of apprehension, dead or alive. So obviously this is different than, the immune system uses different information than you would for apprehending, say, Billy the Kid, but it's still pretty similar. You have the problem, you have a code name of some kind, you have a method of identification and a method of capture. Those are basically the key principles you need for an effective vaccine. So let's just go into the coronavirus here to take a look at how that works. So you'll see that the coronavirus has this, like most lipid envelope viruses, it has proteins on the outside and then genetic information on the inside. And the key protein here to focus on is this spike protein. And if you guys have been reading about it, you'll often hear this referred to as either the S protein or there's a specific portion of this protein called the RBD. That's the part that binds to this ACE2 receptor that allows the virus entry into the cell. And then what happens is once that virus replicates in the cell, it'll eventually bump into one of these things called an antigen presenting cell. That's basically your sheriff, or the thing that's going to be alerted to the presence of the virus. It's going to then grab onto the virus and begin to process that information, alert, say, a T helper cell, which is going to educate a B cell and a cytotoxic T cell. And then that's going to develop antibodies and then start also alerting T cells, which are then going to start destroying the virus. So that's sort of the process that happens. The key thing here is the antigen, which is that method of identification. That's how you know how to tag and identify the virus. And the other thing you need is a way to alert the antigen presenting cell to the fact that this is a virus and not something that actually came from the cell. And that's sort of this code name of some kind. So this is basically what you have in the case of COVID. You have some sort of alias, basically. So the way the immune system actually determines that this is a virus is it looks for a series of chemical signatures in the virus, which it then uses to turn on the immune response. And these have different code names. They're generally called pathogen-associated molecular patterns, which is a real mouthful. And they activate these things called toll-like receptors, but you can just, for the sake of the talk, I'll just call them receptors that get activated. And then there are specific sequences of the protein that it develops antibodies against. These are you typically called epitopes or antigens, and that's what it uses to identify them. And then it uses this method of apprehension, which is a combination of antibodies and T cell responses in balance here. That so you use it to alert each other. So there's a good question there, which I think I can address immediately, which is that how do you keep it from identifying, how do you make it sure that it doesn't attack itself? And so that's because only viruses contain this chemical information and not the rest of your body. So that's how it knows not to generate antibodies against itself. There's also some other more complicated systems called T regulatory cells that make sure you don't attack other things. And we'll get into a little bit more of that here in a minute. So that's sort of the key thing, is that you want to produce effective antibodies against your antigen of interest. That's how you target the virus. And you also want to produce T cells, which really go in and kind of mop up and destroy things. They're basically like hunter seekers that go out and find everything, destroy them. They're also very effective at combating cancer. And the way that you do that is that you basically prime and program naive T cells to either then activate B cells or then become these cytotoxic T cells. That happens by these antigen presenting cells traveling to the lymph node and secreting this sort of complex milieu of different chemical information. And the cell decides to do that by getting activated by this set of chemical information, which is processed by these receptors. And the most important class of these are these toll-like receptors. So these cells, they're called antigen presenting cells. The most common version of them is dendritic cells. They decide whether or not something is foreign or self, basically. Is it a virus or is it part of me? By looking for these chemical fingerprints, right? Basically, they say, are these chemicals coming from a virus or are they something that I know already exists? And they use these receptors to determine that. They say, these molecules could only be coming from a virus because only viruses or bacteria or fungi make these things. And therefore, I know this combination of molecules only exists in a virus. And so that's what you really need. You need these two ingredients to identify and activate either a virus, or if you're trying to develop a vaccine, you can sort of intentionally include these to activate on purpose. You need some combination of these different molecules and then an antigen to ultimately develop the antibody response against. So you need an antigen, which is going to target the protein on the virus's surface, and then these molecules that activate the pattern recognition receptors. And this is basically the chemical code that you feed to these cells, which in turn activates them. So vaccines stimulate a number of these different receptors, and basically all vaccines, all live attenuated vaccines stimulate a whole host of these. And basically every different bug stimulates a unique code. And there's many categories of these, which I won't go into detail, but a lot of my research is interested in sort of how do you find the optimal combination of these or understand what's really critical for them. And with each new pathogen and each new disease, you have to find both what the right code is for stimulating them, what the native disease uses, but also what the appropriate method for activation is. And that changes, right? So sometimes you have smarter pathogens and they'll either find ways to cover up their antigen, or they'll find ways to sort of mask the type of code that will work effectively. And so that will depend on the disease that you're looking for. Eventually, people will usually find effective antigens, but one of the things that will be the hardest to do is actually identify the set of chemical codes that elicits effective long-term durable immunity. So the good news is that for COVID, it looks like this type of response, the type that you need to induce for protection, is going to be relatively easy, and many methods of inducing protection are going to work. So just to cover a couple of the approaches that people have developed. So the one that has been going the fastest in terms of speed is this method of enveloping a piece of mRNA, which is the method that Moderna and Pfizer are using. That piece of mRNA then is injected into cells, and some of that mRNA finds its way into antigen-presenting cells and stimulates them directly, and some of it then produces copies of that viral protein, the spike protein, and so then that protein gets presented by those antigen-presenting cells, and the mRNA acts as a native stimulant. It turns out the lipid coating also acts as one of these codes that stimulates the antigen-presenting cells as well. And those are the two that we heard about in the last 2 weeks with these incredible efficacies. The other four that are really right close behind our development are one by Johnson & Johnson and AstraZeneca and Oxford. And both of those rely on adenovirus, which is a similarly sort of a non-native virus that isn't self-replicating in this case, where you encode the same spike protein, and then the virus itself already has these PRRs and molecules that stimulate because it's a virus. So it finds its way directly to antigen-presenting cells, then activates them, and then they present again that spike protein directly to the rest of the immune system. And the last version, which takes a little bit longer, but is sort of the tried and true method, is to make the spike protein directly and then to add in these molecules directly, these adjuvants in combination, and that's what GSK and Novavax are doing. And it usually takes a little bit longer to make the first versions of these, but then they're very, very scalable, and so they usually make a little bit longer. They usually are able to make them on much larger scale, and the response is usually quite durable. So the last few weeks, as I was preparing this lecture, I've been having to update it because we've seen a lot of really good news. Seriously, really, really, really good news about how this is working. Such good news that I read it in the morning and I had a moment like Scrooge has in, I woke up and went out of bed and I was like, you know, what day is it? Like I was that excited, that level of like my whole life there was like a new lease on life level of good news. And that's because, you know, in the most recent data reports, it appears that the mRNA vaccines that target the spike protein have efficacies that approach 95%. And that's really about as good as you can ask for. Just for comparison, these are the known efficacies for all the vaccines that we've ever developed as a collective human beings. And what you can see is that basically they're nearing the highest efficacies that anybody's ever approached. And what this also means is that because all 6 of these vaccines in development all target the same protein in the same area, it's quite likely that many of these approaches are going to have similar things. The other good news is that, you know, one of the questions is, well, can COVID mutate around this? The other good news is that COVID has this built-in mutation check system, meaning that it basically tries to prevent itself from mutating based on the way that the virus works. So unlike the flu, it mutates much slower, and most of the known mutations at this point, after almost a year of it being in the wild, are not in this RBD region. And so it seems very unlikely that at the rate it's mutating, it's going to find a way to mutate around the antibodies that people are developing. So that doesn't mean we're out of the woods in any sense, because of course, there are still many diseases that don't have effective vaccines. And again, it's interesting to note that we'll have a disease likely now that appeared in 2020 and we developed a vaccine in 2020, which you can contrast to something like tuberculosis, where it appeared in 1882 and we still don't have an effective vaccine for it. So I think there's a lot still to learn about vaccination and a lot to develop here. So these are the questions that my lab is interested in, is what prevents us from completing this list, and how do we develop these things more effectively? And so that's one of the main questions, is not only why is this process so long, but why do we need to dump literally billions of dollars into this process in order to de-risk it? Why is it so risky each step? Why don't we know how to make things or ensure or guarantee between different elements that things are going to work better? And I think that's because studying the immune system is a lot like studying the economy, which is to say that it's fairly easy to tell you what has happened, but it's sort of difficult to tell you to predict what's going to happen, right? So it's difficult to predict an immune response. It's difficult to measure individuals' performance, but it's relatively easy to measure things on aggregate. Macro scale trends are quite reliable. And so at PME, as Jeff mentioned, we like to study sort of molecular and cellular responses. And my lab is particularly interested in how to make vaccines and immunology more predictable by looking at molecular patterns. And that's sort of what we do in this immunoengineering approach. Is that's what engineers are good at. We want to take things, disassemble them, measure them, and put them back together so they can be predictable and they can run like a car, right? That's our sort of our goal. And then we take that understanding and turn it into translation. That's really what we're focused on. And so that's what we take, is that at each of these steps, we understand what's happening at the basic level by quantifying each piece. We then take that basic understanding and translate it into, new approaches to a problem. And then we take those new approaches and bring them to the clinic as new therapies. So to sort of bring this back to these cells, this is really what I'm interested in, is, what are these cells doing and how do they work? Because what I'm about to tell you will probably surprise you. Which is, despite the fact that we've dumped billions of dollars into this process, despite the fact that we've made many vaccines in the world, we don't know how many viruses or vaccines or molecules are needed to activate a single antigen presenting cell, or any antigen presenting cell. We've never observed what the activation of a cell looks like in real time. And then the last thing that's come out of our questions in this nature is, can we change the conversation? How do we manipulate the cells to induce them to make safer and more effective vaccination? We had this idea by asking these two questions first. So just to point out, these are sort of the TLRs and the way they work. You don't need to worry about all the details here, but just the key thing is that they activate by dimerization. That's important. And then they signal through this master transcription factor called NF-kappa B. And that NF-kappa B has to go from the cytoplasm to the nucleus, and we use that a lot as a key signaling factor. So these are the sorts of questions I'm going to go through, kind of 3 vignettes about how we approach these problems. So how many viruses and vaccines or molecules are needed to activate a cell? This is one of the fundamental questions my lab is after. I think it's very important. So we call this determining the number of receptors to activate a single APC. When I talk to other scientists about this, especially at the campus, I talk about it like my Millikan oil drop experiment, because it's very similar in the sense that, Millikan, if you guys remember, was after trying to figure out the charge of an electron, but what he was actually doing was measuring the charge on individual oil drops. And what we're doing is measuring the molecules on individual cells. But when I talk about it informally to my family, and if you're a child like me and you watch this commercial endlessly, I think of it as the how many licks does it take experiment, which is to say, how many licks does it take to get to the center of a Tootsie Pop? Because that is forever burned in my head when I was looking at it as a kid. So the experiment is relatively simple once you understand how to design a molecule to do it. So what we do is we take this antigen presenting cell, and I've drawn it as a cartoon here. We image this NF-kappa B, that's the red in this picture here. We designed a very specific molecule that allows us to observe whether or not it's docked onto this toll-like receptor, the receptor that activates the cell. And we stick a bright, shiny molecule called the fluorophore on the outside of it. And that allows us to count exactly how many molecules are on exactly how many receptors. And then when this NF-kappa B activates, it moves from the cytoplasm to the nucleus, and we can quantify that using a microscope. And as we do that, we can see it move, and we call this the activation threshold. And we can do this on 30,000, 100,000 cells simultaneously using this thing called an image stream, which is basically a way of looking at many, many cells all in all in sequence. And this is the work of a postdoc in my lab, Ethan White. And it's in conjunction with the flow cell operator here at the university. And so we did this by, again, looking molecularly at exactly how to design something. So we looked at the crystal structure of TLR2, figured out exactly how that molecule fit into the groove of this structure, and then found the place where we could stick that fluorophore so that we knew that it would bind irreversibly, meaning that every time one of these molecules bound, we knew it was stuck permanently, and then we knew that we'd have exactly one fluorophore on exactly the end of this site of the molecule. So we knew there was one molecule per one set of receptors, so we could count the number of receptors. And then we get this plot that looks like this, where the brighter the cell, that means there's more molecules, and then we can calibrate that using a calibration curve. And the similarity score shows us the number of the amount of NF-kappa B that's been activated. And so you can see here we have these molecules that stand out, and these are the molecules that are activated. And so we can then go back and calculate. And right now we've just started to figure this out. This is sort of fresh data, but we now know that it's somewhere between 2,000 and 6,000 molecules that are required to activate a cell. And what's interesting about that is that it's much more than the number of molecules that would be on a single virus, for instance. It's probably much more than what would be on a single bacteria as well. And so this is an intriguing question because it means that to activate a response, you would need more than the system would need more than one virus to encounter it. And the question is why now? Why do you need so many viruses or so many molecules to activate an immune response? You would think that it would be much more sensitive. But I think what we're conjecturing is that it has to do with sensitivity, that it needs to have redundancy built in so that it doesn't accidentally set itself off, so that there has to be sort of a signal to noise integration. And we're just learning how all of this works and all of the intricacies involved in it. But this is the first time anybody's ever observed this number. So this is the first time anybody's ever measured this, and we're just learning exactly what that means. So again, to highlight this point, what's happening? So when this vaccine actually encounters a cell, what actually happens to these receptors, right? Again, really nobody knows. What do they look like when you activate them? So this is another question that we've been interested in because, of course, if you wanted to make something round or you wanted the mRNA to be in a different place, or you wanted it to, if you wanted to design something, you would think one of the first things you would want to know is where do the receptors actually live and how do you interact with them? Yet nobody has ever tried to image these things before. And so again, we picked this TLR2 system because we like it and it's easy to work with. And so we started making movies of innate immune responses. So we've been watching TLRs move on cells. And this has been done in collaboration with my colleague, Jun Huang. And really, this is something that could only be birthed out of the CIIC because Jun and I otherwise would be in different departments and there would be no way that we would talk. But now we have a joint student. And so Jun is sort of a master of this. He's been doing it since his postdoc, but he's never done it on this type of cell. And so what he did is, he developed these, it's a special type of antibody where you cut a very specific part of the antibody. It's called a Fab fragment. You then stick a fluorophore on it. That again means that you can image just a very specific part of the receptor. And then he used a very new technique that's only been developed in the last 5 years. It's called lattice light sheet microscopy. What it allows you to do is, like a regular microscope, scans a single beam of light through a cell, but a lattice basically moves the cell through a sheet of light. This allows you to take many more images much faster than normal, which means you can now take basically real-time movies of an entire cell as things are moving on it. And so these are some of the first images we've generated. Each of these yellow dots is a cluster of individual receptors on the surface of cells. And you can sort of see the outline of several of these individual cells here. This is a 3D video. What you're about to see is them moving in real time. So that's an immune cell oscillating and pulsing as it begins to look for and detect pathogens. And what we're finding is that as we look at these, we can track in real time, in seconds, exactly where each of these little clusters goes. using the same type of motion tracking and capture software that you would use that performers use to sort of do motion tracking. So you can sort of create trajectories, you can calculate average and mean velocities, you can look at if these things merge together or come apart. And what you can find from that is that as these cells encounter a pathogen, these things basically act like a set of grappling hooks, almost like, when a ship comes ashore, they cast out lines and basically grab onto this thing and begin to assemble onto its surface and sift out the information in real time as the thing is docking with the cell. And so exactly how those molecules are arranged and exactly how they're interfacing is critical for how the cell understands and then processes the information. And so it shows us exactly how you want to design a vaccine. With the last minute here, I'll tell you one vignette about how these understandings have been leading us to then translate these ideas to improve and change vaccine safety and efficacy. So we were looking at NF-kappa B, and we had this idea about how we could change and manipulate the way that it worked, which could then help us improve safety and efficacy. So again, remember I talked about how these COVID vaccines were activating antigen-presenting cells, and in particular, these adjuvants, some of these later stage development were used to activate these cells. And people often talk about these two important elements of adjuvants, which are that they hold great promise because you can reduce the amount of vaccine that you add. So they're very important. They boost the amount of antibody that you get, which means that you reduce the amount of protein that you add. But then the other problem is that you often get excess inflammation from these. So you can get sort of side effects, which can include headaches, fever, flu-like symptoms. We've all experienced these at some point when we've been vaccinated. Most of these are attributed to a couple of systemic cytokines that come out of the injection site, notably IL-6 and TNF are the two that people mostly regard as influencing these side effects. And so the question is, how much are each of these going to have side effects? And so what we found in studying NF-kappa B and reading about it as we were looking at some of these traditional methods is that, interestingly, the individual subunits that make up this protein seem to have sort of disproportionate ways of influencing this process. And so there's sort of one subunit that seems to do all the good stuff, basically, that leads to protection, and one subunit that seems to do kind of the stuff you don't need as much of. You need a little bit of this, but you don't need that much. And so we had this sort of thought, well, what if we kind of put in a second molecule, what we call a potentiator, that would sort of attenuate or reduce the amount of this subunit that was allowed to cross into the nucleus? What if we could limit the amount of inflammation while still keeping all the good stuff that was coming along with the vaccination? Could we basically make something that was safer and possibly more efficacious? And so these were early experiments that we did in animals. We took a traditional flu vaccine. We added this potentiator. The shorthand is SN50 because it blocks that P50 subunit. And what you can see, and then we added in an adjuvant. This is something that stimulates TLR9 in this case. And what you can see here is that if you do activate it with an adjuvant, you do see much higher levels of TNF. But when you activate it, when you add the potentiator, you reduce those levels back down. So you can eliminate the things related to the side effects, basically reducing them back to zero or near baseline. That also bears out in the way that you measure side effects in animals, which is weight loss. So when we get sort of side effects, we have, we get kind of grumpy and we get a headache. Animals, they do the same thing, but then they don't eat. And so that results in a weight loss. And so you measure weight loss in a mouse, and the more grumpy it is and less happy, the more weight it loses. And so when you add an adjuvant, you'll see it typically loses about 5%. But when we add our potentiator, you'll see that it really only loses about 1% in these cases. And again, when we added this, both with when we potentiated the adjuvants, we got 100% protection in a challenge, which is, again, how you calculate efficacy. Typically, that's for a human. When we talk about a mouse, we just talk about percent protection. And then what we were surprised at is when we took the conventional flu vaccine, the Fluzone, this was from 2018, and we added in just the potentiator on its own, we were able to produce, to boost the amount of protection that the vaccine rendered very, very effectively. So now we're looking at a large grant that actually Jeff is part of, as well as several other people within the CIIC, where we're going to look for thousands of these under a project that'll last for 5 years with the NIH to explore just how far does this concept go? Can we apply this universally to all vaccines? Or is it going to be something that's specific to just a few areas? And so this is something where we've really tapped the knowledge of a huge number of investigators. Jeff is here. Andy Ferguson is another person, Anita Chong, who's in BSD, as well as folks at IITRI who are just up the road. So it's actually across Chicago. But I really wanted to highlight the person who started all of this, Brittany Moser. So I think for all of you who are unaware, every really good idea in a lab starts with one graduate student and she really did all of the work to start this whole concept. And so I think it's really important to acknowledge how a single graduate student can have a huge impact, not only on a program, but on the trajectory of labs, on the trajectory of science in general, and just how important it is to seed and support those individuals as they go along. And that has now given birth to a project that's probably 20 people deep, as well as Brittany Cassidy and Matt Rosenberger, who are now active on this project as well. So I'll wrap up here by just showing you that this works as well to potentiate COVID vaccines. So we've done experiments with the spike protein in the last few months to demonstrate that we can boost the effectiveness of sort of traditionally adjuvanted spike protein vaccines. So again, here's the spike protein, and we're just abbreviating that as S here. So this is sort of what you get as an antibody titer, which is again the thing that best correlates with protection we now know after the last couple of weeks. This is sort of the protection, the levels of antibody that you get if you use CpG, which is again a traditional adjuvant. Coincidentally, this is very similar to the levels of antibody that the mRNA vaccines produce. If you add in our potentiators, this is the protein one, this is a small molecule one, you can increase that by almost an order of magnitude. Again, this is just by adding a simple small molecule that just shifts the way the cell understands the information. And the last experiment I'll show you is not only do we get more antibody, but those antibodies are much more effective at neutralizing the coronavirus. So this is a neutralization assay, which just as you put the antibody in and you see if you can block it from entering a human cell. And so you just keep diluting the antibody down and you see when does it stop being able to block it from entering a human cell. And when we added in our potentiators, we basically ran out of dilutions. You can see it just started blocking, being unable to do that here as we were running out. So it was not only much higher levels of antibody, but the antibodies were much more effective at blocking the virus from entering. So I think these were both very interesting observations that we made as we were looking at how to use these. So, we were developing all this before we got the data about just how effective the mRNA vaccines are and just how good regular antibody titers are. So I think the key things are, you can influence safety and efficacy in interesting new ways. The great news for COVID vaccines are that they're already quite high. The safety margins look great. And it looks like we're going to have some approved vaccines, fingers crossed, in the near future. Of course, everybody has to review the data in great detail. That's going to be the most important part. But to show you what we've been doing, just to summarize, I showed you that we could look at how many viruses are needed to, and molecules are needed to activate a cell. I showed you some of these images about how we could take active movies, making movies of exactly how a cell looks when it's being activated. And then I showed you an example of how understanding these sort of molecular level details could be used to change the conversation, manipulate a cell, and then change the safety and efficacy of even traditional vaccines by potentiating them. And I really just wanted to finish up here by acknowledging the people in my lab, because they're the most important element. They're the ones that do all of the work. I sometimes come up with the ideas. Mostly I act as a cheerleader. And so Xander is the one who does all of the imaging. Ethan is the one who did all of the counting. Brittany is the student I already mentioned, who's really pursuing the potentiator work, but this whole group is really working together as a collective to come up with these ideas, vet them, and generate them. It's a living body that works together collectively to push all of these ideas forward, and they're really the most critical part of the lab. So with that, I'd like to thank all of you for your time, attention, and interest, and the litany of questions I see coming down the pike, and I'd be happy to take the time to answer a few of them. And I shouldn't forget the folks at the Alumni group who were kind enough to invite me to give this lecture. So thank you so much, and I'd be happy to answer any questions.
Aaron, thank you very much for that presentation, going through some of the details of your data, but also at a very understandable level how vaccines work and from a molecular and cellular perspective. We've got about 10 minutes left for questions, and I'll try to walk us through some of the questions that have been posed either before or during the talk. So you mentioned the concept of durability, and questions coming in from Peter, Alev, Paul, Kent, Cynthia, about durability. Does durability mean how long the patient or the subject stays effectively vaccinated? Does it mean the shelf life of the material? Is there a way to predict how durable these vaccines are going to be?
Oh, that's a great question. So when I'm referring to durability, I mean the length of time that the immune response will last, rather than the shelf life of the vaccine itself. That's traditionally how we define durability in the field. And it's a very interesting question because, again, it's something that is very difficult to predict. So it really has to be measured empirically. And so we'll have to see how the durability of these things lasts over time. It's going to be interesting to observe. One of the most interesting things in the piece that I cut out of my talk for time is that some vaccines have incredible durability, most notably the yellow fever virus vaccine, which can last for an entire lifetime. And so the question is, what makes some viruses and vaccines so effective at inducing responses and some that only last for a few years? And this is one of the questions that I'm particularly interested in trying to answer.
Another question coming in from Wendy and from others Sherman, William, one of the features of the flu vaccine that makes it less effective than others is the fact that the flu is mutating so quickly. That's one of the features. How quickly is SARS-CoV-2 mutating, and will that make us have to retool the vaccine after a period of time to deal with it with new mutants?
Yeah, that's a great question. I think one that's actively on everybody's mind. I'm not sure there's any way to answer that definitively, but the sort of good news, bad news part of this situation is that there's a lot of active infection of this disease going on and a lot of sequencing happening every day as we test people. So, and as I mentioned, if you want to look at this more detailed, Trevor Bedford is really the person to look into. He tracks basically both of these diseases on a daily basis. And you can go to his website, NextStrain, to look at mutation rates. But what he's observed is that the mutation rate for COVID is remarkably low, among the lowest known mutation rates for viruses, of sort of respiratory viruses. And so it really seems like compared to most of the viruses that are out there, it's mutating at a rate that's much slower than what we've seen for things like flu. So at least based on what we know in the past, if that's a good predictor for what we see in the future, which is not always true, it seems unlikely that it will be, that the mutations will change. The other thing that we now know is that many of the mutations are not in the region associated with the binding, and that's the region that these antibodies are targeting. And so it again seems like, unlike the flu, which mutates, a lot of its mutations are in the area that the antibodies target. This seems like the mutations that we are seeing are not in the area the antibodies are focused on. And so that's another very reassuring fact about how this will work.
There are several questions on safety. So for example, with the mRNA vaccine from Colleen and Kavita, this is a vaccine technology that's not been used in people before. So how does one judge safety in a clinical study? And then what does one expect based on the biology of mRNA delivery in terms of safety profiles?
Yeah, I think this is a great question. I think there are two important things to note here, which is while there are no approved mRNA vaccines, I think it's a little bit of a misnomer to say it's never been used in people before, because there have been mRNA vaccines that have gone through phase 1 trials. So there are data on the safety of how mRNA works in people for much longer periods of time. And that's part of what what people are talking about when they say there's a backbone of research here. So nobody knows for sure what will happen over time. But as I think, Jeff, you rightly pointed out, the biology of how these work is very different from a live attenuated virus or killed virus. Traditionally, the long-term effects in safety from attenuated viruses come from the fact that the virus is alive and it's actually still inside of you. And so therefore it may have ramifications years down the line. Whereas the mRNA vaccine, it's injected in, it exists maybe for, as far as anybody knows, less than a week before it completely dissipates. And so the likelihood that you're going to have some sort of ramification years down the line seems pretty unlikely, at least based on the way that we understand the biology of how these things work. And again, to sort of note that this is something that's not as well known about vaccines, but there are people, like for instance, in the military who have been vaccinated with mRNA vaccines, a small number, for things like Ebola. And so we have a small number of people who have been tracked for years while they're not approved for widely used for the public. None of those people have ever had any major safety incidences that have occurred for them. That's different than using it and deploying it on a 1 billion person scale. But again, it points to the fact that there's not huge safety issues to be concerned about.
Question from Motz and others about specificity of vaccinal responses. Why don't vaccines cause autoimmune diseases? Even in a patient who maybe is prone to hyperactive immunity and has an autoimmune condition, why doesn't, when the spike protein is taken up into a cell, why doesn't immunity create autoimmunity against the cell that now has spike protein in it?
That's a great question. And Jeff, you know a lot about this too, so maybe you could weigh in as well, because I think I'm not sure anybody has studied or knows the answer to that definitively, in the sense that I've never seen somebody really truly do the experiment. But the explanation that's typically given is that the protein that's introduced either in the transcriptional form or injected is very transient and is dissipated quite quickly. And so most of it is taken up or drained very quickly. And so really it only exists and is presented by those cells for about 24 to 72 hours. And so the likelihood that you would get some response again after that is pretty low. Even for the mRNA vaccines, it's really only expressed for a very short amount of time. So that's the idea of why it doesn't induce autoimmunity. But Jeff, I don't know if you have any other explanations beyond that.
Moreover, there are regulatory immune responses to oneself, so that if there's a gas pedal, there's a brake pedal, and the gas pedal is what Aaron has been describing, how one turns on immune responses. But the brake pedal are different classes of T cells called regulatory T cells that serve to downregulate immunity to self. They even downregulate immunity to a vaccine. So when you're vaccinated, you expand the immune population, but it doesn't expand forever. It expands and then it contracts and leaves behind memory. And that's in part due to this regulatory aspect of it. So maybe just a couple of high-level questions and then we'll finish up. We've got about 3 minutes left. So in terms of immune responses, why has it been so difficult to develop a highly effective flu vaccine? That one comes from Wendy.
Yeah, Wendy, this is a great question. And there's a lot of people working really hard on this in the field. And so I think Jeff already alluded to part of the answer, which is that the flu has sort of two tricks. One is that it mutates a lot, and the mutations tend to be right at the part of the protein that binds with its receptor. And so it tends to mutate very heavily right at the part where it's easy to develop antibodies against. The other is that it can sort of do the second trick where it can wholly swap proteins with itself. So two strains can basically exchange whole proteins, these H and N proteins. So sometimes somebody can get infected and then they can come out with a totally new strain afterwards. Doesn't happen that often, but it's a real pain when it does. So these are sort of two of the reasons that flu has proven really tricky. So the current approach is to develop vaccines that target different regions of this protein that the flu can't mutate around. And there's been a lot of success in that in the last few years. And that's sort of the hope that people have, is that we'll be able to target these other regions of the protein. But that's much more difficult than it sounds, because it's sometimes very difficult to sort of target regions that are what are called non-immunodominant. And so these are some of the other questions that many people in adjuvants are interested in, is how can you sort of design or pick regions to target? So these are questions we're trying to answer.
Yeah, I mean, one last question, which has come up from multiple questioners, but are you going to get vaccinated? Are your children going to get vaccinated? How do you take this personally?
Yeah, as soon as I can is the answer. Yeah, so I think, looking at the safety data that's been published in phase 1, it looks extremely safe. I think there is still data that needs to be done for children. My children are 4, and so the data, it hasn't been tested on children yet. And I think, there'll need to be data run on, there'll need to be tests done on children a little bit younger. And that's traditionally how these things are done, is that they'll start with 18 and then move down in time. Of course, we also know that, those populations are much less susceptible to infection right now after a few years, that's particularly 4 to sort of, it's not clear what age, but, around 12. So I think they're doing things in the right order, but we'll need to see the data on that before people should be authorizing any injection there. But as soon as that data comes out, and I expect that they'll be really safe, because usually if they're safe in adults, they're quite safe in children, then yes, of course. And so for me and my family, I'm sort of counting the days, and, arguing with my mother in particular about making sure she doesn't get other vaccines, because those will interfere with the window in which she can get her COVID vaccine, because, you can't be vaccinated against the flu in 2 weeks prior, and other things. So I'm literally counting the days until it happens. So yes, I think is the short answer. And I think everybody should be eager and willing to do this as quickly as possible.
Yeah, so if I can make a point about that, is one thing that's really interesting about this COVID vaccine Manhattan project is that everybody's been publishing their data. So the level of transparency on the data has been very high. The phase 2s have been published, allowing the broad scientific and medical community to really scrutinize the data very deeply. Finally, just to close, lest anyone take a point of confusion from the last sentence, if there were ever a year to get vaccinated for flu, this is the year.
Oh, yes.
So the health authorities, including the CDC, are really strongly recommending that one gets flu vaccinated, even if you're at home and not exposed very much. And that we want very much to not overwhelm the health system with a bunch of people with flu in the hospital at the same time there are people with COVID-19 in the hospital.
Yeah, just to clarify, what I've been arguing with my mother about is getting her flu vaccine earlier so that she's outside the window of making sure that it doesn't run into the time she might get her COVID vaccine. That's specifically what I've been doing. And then she also needs to get her shingles vaccine. This is probably too much information for all of you, but I'm trying to make sure she times them all out so none of them interfere with each other, and are still working.
Then for much, I'll turn it back over to you, Andrea.
Thank you so much. Hi, everyone. I'm Andrea Hodgman, Associate Director for Intellectual Engagement and Travel, and I'm the Program Manager for the Harper Lecture Series. Thank you, Professor Esser-Kahn, for sharing your knowledge and insights with us, and thank you, Professor Hubble, for being here to moderate. And I'd also like to thank all of you for attending today's Harper Lecture.